So now with the basic background of how atezolizumab and bevacizumab function in inhibiting liver tumor progression, here are some visuals that I put together to hopefully help you gain a better understanding of the mechanisms behind each drug. First, I'll be talking about atezolizumab, which as we know targets the PD-L1 ligand. PD-L1 is a chemical messenger that binds to the PD-1 receptor utilized by many of our body cells, including T, stem, and bone marrow cells. Atezolizumab specifically targets the PD-L1 ligand that is attached to tumor cells, shown in the drawing. Tumor cells can interact with other T cells, resulting in the binding of PD-L1 to the PD-1 receptor, which will hinder the ability of the T cells to replicate or produce cytokines. This ultimately leads to the inactivation of T cells and allows tumor cells to evade our body's immune system. Atezolizumab prevents this by binding to the PD-L1 ligand located on the tumor cell and does not allow it to bind to PD-1 on active T cells. Therefore, the T cells can still function and help remove cancer cells that are present within our body. On the other hand, to better understand how bevacizumab functions, it's important to look at the VEGF pathway that it affects. VEGF binds to specific receptor proteins that are located on vascular endothelial cells. Since VEGF is involved with regulating angiogenesis, or the formation of new blood vessels, in the picture, there is an artery with a magnifying glass that zooms in on the arterial walls. The spindle-shaped object with the dark dot in the middle is an endothelial cell, which forms a single layer on the inside of the artery. The squiggly lines shown below it represent the rest of the arterial membrane. VEGF can bind to VEGF receptors located on these endothelial cells that surround the tumor and promote the creation of new blood vessels in the tumor. This will accelerate liver cancer development by bringing excess nutrients to the tumor and helping remove any toxic waste products. Similar to atezolizumab, bevacizumab binds to VEGF, preventing it from reaching the VEGF receptor. So without VEGF, the endothelial cells will no longer form new blood vessels in the tumor, ultimately disrupting its growth. Thank you.